Today we are going to discuss the functional anatomy of the two main cell lines into which all known organisms can be placed, that of eukaryotic and prokaryotic cells. Last session we talked about the classification of living organisms, and we focused on the three domains as first proposed by Carl Woese in 1978. Now, prior to that, the classic division was the five kingdoms of life, but Carl Woese based his divisions on not macroscopic organismal observable differences, but on the cells themselves, on the cellular metabolism, on the DNA phylogeny, and so forth. And based on this, he divided all organisms into three domains. And it's interesting that when we look at the eukaryotes, we will find 4 out of the 5 original kingdoms in the eukaryotic domain. So we have animals, plants, fungi, and then protists, which includes slime molds, amoeba, algae, and so forth. When we look at the Monera, which was the 5th kingdom of life, we will find that they have now been divided into 2 domains, bacteria and archaea. And he based this on differences in these two domains in terms of DNA phylogeny, in terms of their metabolism, in terms of the way they undergo replication, transcription, translation, and so forth. Today we're going to even further simplify these 3 domains or condense them into 2 main cell lines, that of eukaryotes and prokaryotes, which includes both bacteria and archaea. So let's look at eukaryotes for a moment. Eu is Greek for true. Karyos is Greek for nucleus or nut. So eukaryotes have a true nucleus. The DNA is within a membrane-bound region within the cell. On the other hand, prokaryotes, pro is Greek meaning pre or before. So prokaryotes have a pre-nucleus. That is, they have DNA in a nucleoid region, but it is not contained inside a nucleus. Here we look at a prokaryotic cell, and you can see that if you look in the interior, and this is based on a transmission electron micrograph, you can see that the DNA is in a nucleoid region, but again, it is not contained within a nucleus. There's also an extra chromosomal small piece of DNA called a plasmid, and then you can see ribosomes throughout the cytoplasm. And then you notice that the most amount of complexity is on the outer surface of the cell. We see fimbriae, we see flagella, apellis, and then this cell also has a capsule. And so for bacteria, often, or archaea as well, often what is being interacted with in a clinical lab will be the outer surface, whether it's differential staining or immunological techniques or other processes to determine what type of infection a patient has, for example. So in looking at prokaryotic cells, remember that they are always unicellular. So archaea and bacteria are always unicellular. They're quite small. So the average size of a prokaryotic cell is only 2 to 8 microns in diameter or in length if we're looking at a bacillus. So the longest bacillus cells that we'll see in general are only 8 microns in diameter or in length. And a micron is 10 to the negative 6th of a meter, so it's 1/1,000,000th of a meter a micron is. So remember, these are quite small organisms. Of course, as we've mentioned already, there's no nucleus and no organelles. In contrast, as we look here at a eukaryotic cell, we see that there is quite a bit more complexity in the interior of the cell. You can see the nucleus, you can see quite a few membrane-bound organelles, the mitochondria, the Golgi, the endoplasmic reticulum. However, this is an animal cell, so you're not seeing a cell wall. You're not seeing quite as much visible complexity on the cell surface. Though we know it's there, it's just not as visible. Remember that eukaryotic cells can be both multicellular or unicellular. So multicellular, of course, includes plants and animals, whereas unicellular, yeast are often unicellular. Fungi can be either, as can algae. Protists, protozoa, are always unicellular organisms. Eukaryotic cells have a nucleus and numerous organelles. They are also quite a bit larger than prokaryotic cells. The range in sizes that we see in a eukaryotic cell is generally 10 to 100 microns in diameter. Some are quite a bit larger. Some of the protozoa can get quite a bit larger than that. And even some of the human immunological cells, like macrophages, are also larger than that. Then there are also some that are smaller. Yeast are generally smaller than 10 microns. And even the human red blood cells are also about 8 microns in diameter. Today we're just going to discuss some key differences between the cell lines, and this includes size, structure of the cell wall, function of the cell membrane, the nucleus and other organelles, arrangement of the DNA, the ribosome, and cell division. So let's talk first about the prokaryotic cell wall. It's a complex, semi-rigid structure. It's responsible for cell shape. So last time we talked a little bit about different shapes of bacteria, the bacillus, the rod shape, the coccus, the round, the curved vibrio, and the spiral spirillum. And this is all based on the cell wall. So different bacterial cells will have different shapes based on their cell wall. The cell wall protects the cell from the outside environment, prevents osmotic lysis. If it's in a hypotonic environment, it will prevent the cell from bursting. The prokaryotic cell wall is made of peptidoglycan in bacteria and pseudomurine in archaea. Now, we're not going to talk very much about pseudomurine today, but just know that it's quite similar to peptidoglycan. It is also composed of a sugar or carbohydrate glycan-like component, but it's different disaccharides. And it also has a peptido component as well, so a component that's made of amino acid crosslinks. So let's just break down what that means here. So peptidoglycan, first of all, is a polymer of repeating disaccharides in rows. So here we can see N-acetylglucosamine and N-acetylmuronic acid, or shortened to NAG and NAM. And this will be a repeating disaccharide, hundreds of thousands of units in a row. So we can see that here, represented here, NAG, NAM, NAG, NAM. And then we see a cross-link of amino acids, polypeptides, 10 going one way, and then 4, a tetrapeptide forming a cross-link. And so this is one layer of peptidoglycan. But if you look at a gram-positive cell, you're going to see layer upon layer of this, forming a very, very thick and strong protective wall around the cell. As we move on to prokaryotic cells, we see a little bit different structure. We see a lot more complexity in the interior. And then here we see a key difference between animal cells that don't have a cell wall and plant cells that do. And just keep in mind that algae and fungi will also have a cell wall. Let's just talk briefly about the eukaryotic cell wall. There is no peptidoglycan in the cell wall. So eukaryotic cell walls are primarily composed of carbohydrates. So in plants and algae, that carbohydrate is cellulose. So it's repeating glucose or glucan units. Whereas in fungi, it's composed of chitin, which is an N-acetylglucosamine polymer. And so notice in both cases, it's carbohydrates. There is no peptide linkage. There's no peptido. There's just the glycan component. And so just keep in mind, peptidoglycan is one of those components of a bacterial cell wall that can be a key target of antibiotics. And so the first antibiotic, penicillin, targets peptidoglycan, and it specifically targets the peptide linkages in the tetrapeptide cross bridges. Let's move on to talking a little bit about DNA. And so when we look at DNA in prokaryotes, again, here we can see an electron micrograph. We can see the nucleoid region here. But even though you see this massive DNA here, and for example, in E. coli, the entire genome is a little over 4 million base pairs. But in a bacterial chromosome, generally speaking, it's a single circular thread of double-stranded DNA that contains all of the cell's genetic information, or in other words, its genome. The DNA is often attached to the cytoplasmic membrane, and this aids in that binary fission that we're going to talk about in a moment. There are often also extrachromosomal genetic elements, such as plasmids, and these are usually only a few thousand base pairs in length, and they will also be circular, and they'll carry non-crucial genes, such as, for example, antibiotic resistance genes, or sulfanilamide resistance genes. And so sometimes these can be quite a big problem in the medical community, because bacteria not only have these plasmids, but they also have mechanisms for sharing them with other bacteria. So here we see Apellis here, and that is one of those components that can be used to share small pieces of DNA with other bacteria in a chromosomal or in a genetic transfer, a plasmid transfer. In eukaryotes, we see quite a different structure. Here we have the membrane-bound nucleus here. We have the nucleolus where the DNA is condensed, and so that contains the DNA. The DNA is generally complexed with histone proteins. So here we have the double-stranded DNA, and then it's wrapped around histone proteins. And what this will do is allow the DNA to become very, very highly condensed into chromatin. And then during mitosis and meiosis, the chromatin even further condenses into these chromosomes. And so chromosomes, there'll be two copies, so there'll be paired chromosomes in each cell. And so this system of histones, chromatin, and chromosomes allows a very, very large amount of DNA, very long pieces of DNA, to be condensed into a very small area. Let's just end with the cytoskeleton and cell division. So cytoskeleton proteins are involved in cell division, maintaining cell shape, growth, and DNA movement. And so in eukaryotes, we're familiar with the microfilaments, the filaments, and the microtubules, and these are all involved in mitosis as well as meiosis. And so eukaryotic cells that are not undergoing meiosis will divide by mitosis. And earlier, this was thought to be the primary role of cytoskeleton proteins because during mitosis and meiosis was when we would see them in action. And it was even thought that prokaryotic cells didn't really have very much in the way of cytoskeleton proteins. But we found that analogous proteins are indeed found in prokaryotes, including the FITC, the MREB, and others. And so these are important not only in the division of prokaryotic cells, which we don't see them playing as large a part of a role in the division during prokaryotic cells, but they do play an important role in movement throughout the cell, movement of not only DNA, but other components, other proteins, other larger components throughout the cell. They maintain cell shape. They help the cell grow properly. And so prokaryotic cells don't divide by mitosis, they divide by binary fission. And here we can see a prokaryotic chromosome in complex with the plasma membrane. We're seeing duplication of the chromosome. And as the chromosome duplicates, the cell moves into septum, and then there's complete division between the two cells. And here we can see a scanning electron micrograph of a chromosome, a bacterial chromosome, during replication, and so we can see the replication forks, and as it forms its its second copy of the chromosome, this then needs to be resolved at the replication forks, and you'll see resolvation into two circular chromosomes. And as that is occurring, the cell itself is also dividing. And so bacterial cells such as E. coli divide about every 20 minutes. Is their generation time. So just think of how quickly that DNA is being replicated and how quickly that cell is dividing. About 4 million base pairs of DNA is being replicated within 20 minutes in E. coli cells. So it happens very, very rapidly. So we're not going to talk in detail about all of the eukaryotic organelles today, but just know that they include the nucleus, the ER, the smooth ER, and the rough ER, which includes the ribosome, Golgi complex, lysosome, mitochondrion, chloroplast, peroxisome, and centrosome. And so our next session will be spent talking in a lot of detail about all of the different eukaryotic organelles, their function, and their structure. Now I'd like you to just take a few minutes and think about all of the differences that we learned today about eukaryotes and prokaryotes. And if you have a piece of paper or a pen, maybe you can even jot down a list and perhaps just pause the video for a moment and write down the list and then come back and see how many you've remembered. So let's just go over these quickly. In eukaryotes, we have paired chromosomes inside a nucleus, whereas in prokaryotes, we have only one circular chromosome, generally speaking, and it's not enclosed in a membrane. For eukaryotes, the DNA is wrapped around histones, and there are no histones in prokaryotes. Eukaryotes have organelles, including the nucleus, mitochondria, and so forth. There are no organelles in prokaryotes. Eukaryotes have 80S ribosomes, whereas prokaryotes have 70S ribosomes. But of course, as we talked about, eukaryotes do include 70S ribosomes inside the mitochondria or the chloroplast for plant cells. There are polysaccharide cell walls if present, so cellulose for plants and algae, and then chitin for fungi. And then peptidoglycan cell walls in bacteria and pseudomurine cell walls in archaea. And then finally, eukaryotes divide by mitosis or meiosis, whereas prokaryotes divide by binary fission. So I will see you next time.